All right. Hello, everyone. Nice to meet you. My name is Garrett. I am one of the learning assistants for your class this semester. And I know that we aren't going to be meeting for class, but I am, as my title suggests, here to assist in your learning. And that means sometimes, like in this video, I'll be here to help you solve problems that you will wink, wink, probably find on an exam or two. Um, and you are absolutely welcome to, and I would love to see you in my review sessions and exam reviews, or anytime on campus if you are so inclined. And you can learn more about me and get access to my links and emails and stuff, as well as for the other learning assistants, Yekta and Vaishnavi, um, here in the LA Resources tab on Blackboard. Hopefully you've been able to find your way around, and I assume you are finding this video in the week 1 part of Learn Here course content. But as I said, I would like to solve a problem with you. Here at the beginning of the semester, we are in chapter 2. So mitosis, meiosis, some really fundamental foundational stuff for genetics. Excuse me, this should be a review of maybe AP Bio or 213 if you took it here at GMU, or any future class. But it's one that, it's tough, and it's no shame to need to draw it out. That's how I still do it. This is my fourth or fifth semester with genetics, and I still draw it every time. I think it's the most effective way. And so that's what we are going to do together. I very strongly, hopefully, or excuse me, very highly recommend that you watch Dr. Gantar's videos on this before, so that you know your cell cycles and all of that very fun stuff. But I am going to share with you some of the tips and tricks that I use to navigate these questions. Because like I said, wink, wink, you're going to find this question on at least one of your exams. This is a very important question. Being able to identify the amount of genetic information that's in a cell at a particular stage in that cell's life cycle. And on that note, let's get started. So how many chromosomes and DNA molecules are present in each cell during the following stage of meiosis? So we are in meiosis. What is meiosis? And what does it contrast to? So the two big M's that we think about, and they're the title of this chapter, is mitosis and meiosis. The point of mitosis is to start with a cell, and it will create two daughter cells that are identical. And they're called daughter cells because they come from a parent cell. And they should be identical to each other. Meiosis, however, is all about taking that parent cell and creating 4 unique daughter cells. Still daughter cells, but we want them to be unique from the parent, because what's going to happen is that these daughter cells, their genetic information will combine with the genetic information from another organism to form a zygote, and we know that that will divide, blastulate, and become an entire organism. So that's what we're doing here in meiosis. We are trying to create that condition. And we have, like I said, to start off with a parent cell, and that's what we see over here. There are a few things that I notice immediately with this. Oh, did I not erase that? I actually spent like an hour and a half doing this video earlier, which I should not have done because an hour and a half is way too long, and I messed up a whole bunch, so take 2. So this cell right here, we see that it's in G1, and I did just mention it earlier that our cell looks different at different stages in its life, and that makes sense. If you have, for example, an endocrine cell, it is, most of the time going to be doing its endocrine stuff. Let's say it's an endocrine cell in the cortex of your adrenal gland. It's going to be producing cortisol, androgens, aldosterone, that endocrine stuff. But for a certain subset of its life, sorry, bonk, for a certain subset of its life, it's going to have another like little mini cell cycle where it's doing some non-endocrine stuff. We call this interphase, and there are some really important things that happen in it, all this stuff. And in that we have, you should hopefully have watched Dr. Gontar's video. We have the G1 phase, and I think G for growth, because this is where our cell, like for example, if this is a new endocrine cell, it started out as a little baby, and it has to physically grow. So it will undergo its growth in G1 phase, then it will move on to the synthesis phase, or excuse me, the S phase, sorry, which I gave away, means synthesis. It stands for spelling, synthesis. What are we synthesizing? This is talking about the DNA, of course, and that is what we're here for in genetics. We're learning about DNA. And when I think of DNA genetics, I usually think of, these little squiggly guys, those chromosomes. But what is a chromosome? What is, do we just have these little X's hanging around in our cells? Not really. What we do have, or this is what a chromosome really is. If we take our chromosome here on the left and we zoom out, let me find that really fast, sorry. And we zoom out, excuse me, if we zoom in, forgive me, we see that our chromosome is just a lot of fiber, a lot of wool that is really tightly coiled together. And if we zoom in on the wool, we see that it's actually a coiling of an even smaller thread. And if we zoom in very, very far on this nice small blue thread, we see that it's actually our nice DNA double helix with our nucleotides. And these are our A's, T's, C's, and G's that are the actual information that is transmitted. So when you're looking at your X's and even your little lines over here, that's what you're actually looking at. You're looking at very highly condensed DNA double helix. And on these X's and on these lines, we have our information. What is the difference between an X and a line like we see here? Well, that harkens back to the cell cycle, especially the synthesis phase, the S phase. And oh boy, you might hear some noise, and I apologize for that. We have the synthesis phase, and that's because our, during the majority of our cell's life cycle, oh my gosh, one moment, please. Okay, sorry if you heard a bunch of dog barking, that's annoying. Where was I? Oh yeah, synthesis phase. So during the normal life cycle of our cell, our DNA doesn't look like these nice, cute little X's. They're actually only a small amount of genetic information, still condensed at some points and unraveled at other points, but we can think of it as only being comprised of a single, one of these units. And this unit is what we would call a chromatid. And this chromatid, because, actually no, so we have a bunch of genetic information in our cells. Get out of here. But we don't just have one piece of DNA. I'm sure this would look familiar to you. This is what we call the human karyotype. It is a representation of all the genetic information that we have in our cells. And we have these chromatids here. We have this one here this is our 6th chromosome. We have 23 pairs. All of the DNA in our body usually sits in the nucleus, and it's all this just wool, thread, spaghetti, whatever you want to call it. But at certain points in the cell cycle, they condense into distinct units, which we see as chromosome 12, as chromosome 17, as chromosome 3. And yeah, these are the chromatids that we see. But the chromatid at this point in the cell cycle represents all of the genetic information for that particular portion of the genome. Let's say that this is chromosome 14, for example. This single chromatid, this single segment of DNA, represents all the genetic information that we find. Oh my gosh. Lovely. For, sorry, this is chromosome 14. We consider this chromatid to be a chromosome in its own right. Hello, sorry about that. Just a little distracted. Where were we? Oh yeah, talking about chromosomes. So we have these things called chromosomes, and chromosomes are the condensed amalgamation or collection of all the genetic information that we have in our cell. But these chromosomes don't always exist as our nice pretty little X's. At some points, they're only comprised of these single little strands. However, in the synthesis phase, these little strands are going to undergo DNA replication, which we'll learn about in chapter 12. But what happens is that DNA replication, this strand of DNA here, the chromatid, which is a chromosome in its own right at this point, is going to duplicate every single one of its nucleotide bases, hopefully be a perfect copy. That would be nice, so we can have all the same instructions. And it's going to synthesize a perfect copy of itself. And this chromatid now has a sister, a twin sister. And we call these, relative to each other, sister chromatids. And hopefully you have once again seen Dr. Guntar's video, so this shouldn't be new, but these sister chromatids are perfectly identical to each other. And what would you say, now that we had this thing that was a chromatid, or excuse me, a chromosome in its own right, and these two usually join together. This one is this, and this one is this one. So they usually join together and they are connected by a centromere, also cohesin proteins, but you don't have to worry about that for now. So what do we think? Is this structure two chromosomes? Because this was a chromosome in its own right, and now there's another one right next to it? I wouldn't say so, because these chromosomes aren't just like little bars that our cell wants to have. They have specific instructions along them, you know, the A's, T's, C's, and G's that we use. And because this should be an exact replica of this, this provides no new information, no new instructions that our cells can actually use during its many vital processes. So we consider this not to be two chromosomes, but a single one. However, it is made up of two chromatids that are sister to each other. This chromatid is sister to this chromatid, just as this chromatid is sister to this chromatid. And an analogous term for chromatids is a DNA molecule. I like to say chromatid because I'm cheeky, but they're also called DNA molecules. So we have this single chromosome that is made up of 1, 2 DNA molecules, or 1, 2 chromatids. And that's what this problem is all about. So let us get started on that. Actually, there are a few things that I want to articulate beforehand because we're going to talk about some genetic variation in the future. And these sister chromatids, they join together to form single chromosomes for some of the cell cycle, particularly this interphase. And let's say you have an entire chromosome right here. And it's made up of 1, 2 sister chromatids, but it is itself just a single chromosome because the two sisters are identical. They carry the same information. Excuse me. So they are considered to be just a single chromosome. And let's say that we inherit this from one of our parents. We are humans, Craniata bilateria. We are diploid organisms. what is a diploid? That's kind of an alien sounding word. I like to use etymology and break down alien sounding words so that they are more comprehensible. Di is a Greek prefix meaning two, and ploid means folds. And when we're talking about folds, we're talking about these X's because they're kind of like drawn in a fold. So when we say two folds, we mean two chromosomes. Humans are diploid because we have two chromosomes that represent genetic information for the various portions of our genome. We inherited this one from our mother and this one from our father. We have two chromosomes. Let's say that this is chromosome 14. We go back to our little karyotype guy here. That's exactly what we're looking at here. We inherited this one from our mother and this one from our father. We have two chromosomes that represent the information for that specific portion of our genome. And this is important for genetic variation measures as well, because these chromosomes are what we call homologous. Homologous, that's another big word. But once again, let's break it down, and we see that homo means the same or similar, and log, like you keep a log book, refers to information. So homologous chromosomes have the same information. For example, at this point on the chromosome, you have, at this point on the chromosome, you have the gene that codes for the length of your femur. And at the same point on its pair, you have the one chromosome here, and then you have its pair right here. At the same point, the same location on that chromosome, you also have the gene for femur length. And these locations, oh, what happened? Oh no, that's not good. I don't like that. These locations we call a locus, or the plural is loci. So at this locus, this specific point on that far down on the chromosome, you find the gene for femur length. And similarly on this one, at the same point, the same length along the chromosome arm, at the same locus, you also find the gene for femur length. And you will find that all along these chromosomes, you have the same genes. And that's what it means to be diploid. You have two chromosomes that represent a portion of your genetic information. And as we saw in our human karyotype, we have 23 such pairs, 23 pairs of homologous chromosomes. But just because they have the same information, they contain the same genes, doesn't mean that they are like exactly identical. So what we see, for example, considering our nice red, bright red chromosome here, we have this femur length gene here and here, because these are two sister chromatids. They're sister to each other. They are perfectly identical. So you have this nice mustardy green for femur length here and femur length here. And then if we, but if we consider this maroon chromosome here, let's say we have also femur length gene at the same locus. Pretend that these are the same size of chromosome. They should be, because they're if they contain the same information, they should be the same length so that they have the same number of nucleotide bases, and therefore the possibility of having the same information. But let's say that you also have femur length gene here and here, but this color is different. Oh my gosh, what's the deal with that? Well, we have to keep in mind that genes are nothing more than our sequence of nucleotide base pairs. So let's say that this sequence here, AATCCG, most genes are much longer in length than that, but let's pretend that this still codes for, or this codes for femur length gene. And we have this that also codes for femur length gene. and actually it would not be that because A can't pair with A, but let's say that it would be AACCCG. That's right. Let's say that this worked as a femur length gene, and this also worked as a femur length gene. They are slightly different, like different flavors, but they still code for that same gene. Having genes that are represented by different sequences, but they still code for the same gene. These are called alleles. They are different variants of the same gene. This light yellow allele codes for, let's say, a femur length of 0.7 meters. And this one codes for a femur length of 0.75 meters. It's still femur length gene and it's pretty similar, but these measures of genetic variation come in handy in later phases, such as metaphase 1. And with that very fundamental stuff in mind, I think we should get started. So remember, we need to keep in mind our chromatids that are sister to each other, but also keep in mind that we have chromosomes that are homologous to each other. And let's remember, twin sisters are identical to each other, but homologous chromosomes, while they have the same genes along them, you could have different variants of that gene. And it could be that, you have this situation for femur length gene, but for the gene that codes for, I'm so bad at thinking of examples, the diameter of your, oh God, your glenoid cavity, I guess, glenoid cavity diameter. These could be G, 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 G. There doesn't have to be a difference in the sequence. They just happen to have, it could be that this chromosome, which is homologous to this chromosome, has the same allele on it. But it could be that they have different alleles. It's, we have a lot of genes. There's a lot of base pairs in our genome, several billion. So there's a lot of opportunity for variation. And now getting on with it, we were talking earlier about the synthesis phase. And that is where that is the point where our chromosomes, which we see on our handy little drawing here, our chromosomes, we have big red and little red, big blue and little blue. And they are each chromosomes in their own right. This is in G1 phase, which precedes the S phase. So when we move into the S phase, we have synthesis. And already, just as a little side note, we know what kind of organism this cell is. Oh, also, I forgot to articulate that at any point that I ask a question, I invite you to pause the video and try to answer it with me, have some interaction in quotations. So what kind of cell would this be? There are a few clues. I would say that, it is a eukaryotic cell for two reasons. We know that bacteria have circular DNA. They have little, what we call nucleoids in their cells. Their cells don't have membrane-bound organelles. They kind of just float around in the cytoplasm, if you consider a bacteria with a flagellum and some cilia. Their DNA and all of their organelles kind of just floating around. They don't have membrane-bound organelles like our cells do, where we have our nucleus with some chromosomes inside, and we have our nice, beautiful, wrapped endoplasmic reticulum and stuff like that. So the fact that this cell here has a nice nucleus containing DNA. That's a very good hint that it's not a bacteria. And also, like we mentioned, bacteria have circular chromosomes, and these chromosomes are linear. So we can infer that these are eukaryotic cells. Anyway, with that out of the way, and also actually we don't have to assume that. Anyway, so synthesis phase, what we're going to do is take our chromosomes, which are made up of just a single chromatid in G phase, we are going to undergo DNA replication and duplicate them such that every chromosome is now comprised of 1, 2 chromatids. And what is the relationship of this chromatid to this chromatid? Exactly. It is a sister to that chromatid. What's the relation of this chromatid to this chromatid here on the right? It is also a sister. Perfect. And so we essentially, in replication, we duplicate the amount of genetic information we have. So in G1 phase, I almost forgot to start there. We see 1, 2, 3, 4 chromosomes. 4 chromosomes, and each one is made up of just a single chromatid, a single DNA molecule. And we can see that. So that's part 1 done. But in S phase, as we see, we duplicate. We replicate the amount of genetic information we have. So every chromatid will replicate a sister so that every chromosome is now comprised of 2 DNA molecules. So we have little blue that will duplicate, will make a sister for itself. And we have big blue that will do the same. So let's draw that in our guy. Little blue is now made up of 1, 2 chromatids, and big blue is made up of 1, 2 chromatids. And the same thing will happen to little red here and big, oh no, right, please. And big red here, is my pen dead? Are you serious? That'd be stinky. No, it's fine. What's up, pen? Okay, I don't know what happened. Oh yeah, there, okay. So little red also jumps in. It has duplicated its sister, or it has copied itself to make a sister. And big red has done the same. So we can, just do some counting. We see that there's 1, 2, 3, 4 chromosomes. Nice. I'm also going to write this in a different color. 4 chromosomes and 4 DNA molecules for G phase. But in S phase, we have 4 chromosomes. However, each one is made up of 2 chromatids, 2 DNA molecules. So we can do 4 times 2, or we could count them. We have 1, 2, 3, 4, 5, 6, 7, 8 DNA molecules or chromatids. Woo! And G2 phase is the same. And if you would like an explanation why, please refer to Dr. Guntar's video. So that is S phase. And after S phase, we undergo the G2 phase, which is pretty much just setting up for mitosis or meiosis. And then we move into prophase, which I will represent with the letter P. And 1 second. Okay, prophase. These phases can, there's a lot of them and they can be easily confused. So let's try to keep them track by knowing, once again, the etymology of their names. Pro means before. So it is prophase. What happens in prophase is all the stuff that happens before, like the main objectives of mitosis and meiosis, where we create our daughter cells by splitting stuff apart. So we're just setting up. We're doing all the stuff before, like the big cool stuff of mitosis and meiosis. And what happens in prophase is that our nucleus that we saw has almost entirely dissolved. That's one of the big things. We need to get our genetic material out of the nucleus so that we can actually do all of the separation and alignment and stuff that we need to in mitosis and meiosis. So we are going to have our exposed chromosomes for the first time, and these are going to finally be condensed, like entirely condensed into our nice, neat chromosomes. It's no longer all of this spaghetti or thread nonsense that's just all taking up all the space in our nucleus. It has condensed down into these nice little chromosomes. And also our spindle fibers will form, and these will come into effect in later parts of the cell cycle. So we have our big red, big blue, little blue. And we can count once again. We have still 1, 2, 3, 4 chromosomes. Each one is still only comprised of 2 chromatids, 2 DNA molecules. So we have 4. I'm going to use black. I will try to be, I was not consistent with the colors in the last video, and it was a huge mess. So I'm going to be better about it this time. And also I don't write 8 like that. So prophase sets us up for metaphase. And meta means center. Meta means middle. And metaphase is where our chromosomes align in the middle, in the center. And so during metaphase, if it's possible, or you'll see what I mean when I say if it's possible, chromosomes will align in the center in their homologous pairs, preferably. Remember, we were talking about homologous pairs. Homologous, they have the same information. And in order for them to have the same information, they should be the same length, you know, because you don't want, chromosomes are already condensed enough. So you wouldn't want all of your information condensed more than your partner. You need your chromosomes to be the same length if they're going to align properly. And another point to make is that when we're doing problems like this, we only see like 3 or 4 chromosomes because in reality, humans have 23 and that'll just be a mess to draw. Owls have like 82 and different organisms have different numbers. It gets to be a mess. So that's why you only see 4 chromosomes here. And as I said, our homologous pairs align in the center. So who is big blue going to line up with? Big red, that's right. And conversely, oh wait, no, yeah. Little red will line up with little blue. And that's metaphase. They align in the middle, in the center. But then we move on to one of the most important phases. Oh, let's not forget our spindle fibers. Here, our spindle fibers also attach to the, as we articulated earlier, the centromeres, actually the kinetochore technically, but don't worry about that for now. Our spindle fibers will attach to the, no, I didn't mean to do that. No, I didn't mean to do that. Will attach to the centromere. And when this spindle fiber shortens, kind of like you have an elastic stretched out, it will try to shorten back up to only be like that long. What that's going to do is it's going to pull your chromosome apart, or not apart. It's going to pull it toward the spindle fiber. Whoosh. It's going that way. It's going to be pulled to the pole of the cell. It's no longer in the center, it's at one of the 2 poles. And that is what anaphase is. Anaphase is when our spindle fibers actually contract and pull the chromosomes apart from each other. Oh, I forgot to mention that. So we have our spindle fibers attached to the kinetochores and also big blue and little blue being pulled. So anaphase is important because what we're trying to do is take our parent cell and reduce it. We don't want our diploid parent cell, we don't want to put like 2 copies of a chromosome into our gametes, and that's what meiosis is all about. Meiosis happens in our germ cells, and they produce gametes. So it happens in our germ cells and should produce 4 gametes, because these gametes need to combine with another organism's gametes in order to create the zygote. I think I'd said that in this recording already. They will combine to produce a diploid zygote. So what we need to do is reduce the amount of information that we have, because we start off diploid. We have, for chromosome, let's call the big chromosomes chromosome 1, because we have the 23 chromosomes. So let's say that this organism only has 2 chromosomes. We have the big ones, those are chromosome 1, and the little ones are chromosome 2. We have 2 copies of chromosome 1, the big ones. We have big red and big blue, and we have 2 copies of chromosome 2, little red and little blue. So what we need to do, or that's what we start off with, but in order to go from germ cell to a more simple gamete, we need to reduce our ploidy. Remember, we started out diploid, 2N, di means 2N, and we need to go to a single N. We need to have haploid gametes. And haploid, I think of as half-ploid. And that's just relative to the original parent cell. Animals are commonly diploid, but plants can have crazy numbers of ploidy. For example, you could have a tetraploid plant. Tetra as Latin means 4. So it doesn't just have big red and big blue. It has big red, big blue, big yellow, and big purple. And when that undergoes meiosis, it will be split into haploid 2N. And this is haploid, this 2N, relative to the 4N that it started out as. Just a little note. So what anaphase is all about is reducing the amount of genetic information that we have, and we need that in meiosis so that we can create daughter cells with an appropriate ploidy. So anaphase, we reduce our genetic, or we simplify our genetic information by separating homologous chromosomes from each other. We have to separate big red from big blue. It's so sad. Oh my gosh, it's terrible. It's so sad. And that's how we simplify the genetic information at this point. We are in meiosis 1. I didn't articulate that. And the only way we can simplify this genetic information is by separating the homologous chromosomes from each other. They gotta go away. And what's going to happen is that in anaphase, the spindle fibers separate homologous chromosomes from each other, just like we said. And then, so once they're separated, we move on to telophase. That was terrible. We move on to telophase. And in telophase, we form a cleavage furrow. Oh, let's remember, so our blues were pulled to the left side. So we have big blue and little blue. And our reds were pulled to the right side. So we have big red, little red. And in telophase, what starts to happen is that we have proteins at this point here on the cell and this point here that cinch the cell and try to split it in half. And I remember this is called the cleavage furrow. I think of it as like someone takes a cleaver and like chops down into it. I'm sure you can think of another memory device for why it's called a cleavage furrow, but oh yeah, we haven't been keeping up with the chromosomes and DNA molecules. Let's do that before we move on. So metaphase, once again, I think it's important to draw all of this out. I think it's the most effective way to keep everything straight. And that way you don't have to like guess at the math. You can just simply look back at your pictographic representation of meiosis or mitosis and literally just count chromosomes and DNA molecules. So we see in metaphase that we have 1, 2, 3, 4 chromosomes. And each one is made up of 2 DNA molecules, so 2 times 4. We could also count them 1, 2, 3, 4, 1, 2, 3, 4. Or sorry, 1, 2, 3, 4, 5, 6, 7, 8 is what I meant to say. So 8. And then in anaphase, we still have within the same cell 1, 2, 3, 4 chromosomes, each one comprised of 2 DNA molecules. Then in telophase of meiosis 1, I would still consider this to be a single cell. You know, they haven't physically split into 2 separate entities. So I would still say that we have 1, 2, 3, 4 chromosomes. Each one is still comprised of 8 DNA molecules, or 8 chromatids. So our numbers haven't changed throughout this. Oh, actually, yes. And for some reason, remember we have our 4 and our 8. There's another thing that happens before meiosis 1 ends and meiosis 2 begins. We have a cytokinetic event. This is the final stage of meiosis 1. It's called cytokinesis. That's another alien sounding word. Cyto means cell, and kinesis, like kinetic, means movement. So it is literally the movement of cells, because we have this cleavage furrow starting to form. And after they actually split apart, once the cleavage goes all the way down and they meet in the center, the 2 cells are going to move. The cell moves. And that is our cytokinesis. And the result is 2 separate daughter cells. And remember, we had our blues on the left. So we have big blue and little blue. We have our reds on the right, big red, little red. Pretend they're the same size. And this is the end result of meiosis 1. What we've done is create how many daughter cells? 2. And are they identical or unique? They are unique already, even at this point, relative to the parent, because the parent had half red and half blue DNA, but this cell has 100% blue DNA. This one has 100% red DNA. So that's already different, unique relative to the parent. So these are the end products of meiosis 1. And for some reason, cytokinesis was not given its own little section. That's okay. Cytokinesis 1, pretend there's an either kinesis 1. It's curious that it doesn't have its own section, because this is where the numbers actually change for the first time in a while. Because we had 4 and 8 in telophase. Oh, I erased it just to write even less attractive numbers. What's wrong with me? There. 4 and 8 here in telophase. But when we look at the number that exists in each cell, by cytokinesis, these cells are their own cells now. So we can only count 1, 2 chromosomes. And each chromosome is made up of 1, 2 chromatids, 4 total. So the numbers finally change here in cytokinesis 1. And like I said, these are the end results of meiosis 1. And let's focus on just a single one of these cells. Let's choose blue. Let's choose blue, and we will investigate what happens to blue in meiosis 2. Because these cells, the blue result of oh my gosh, please let me select it. You're a menace. So this is the final result of meiosis 1, and it will go on into meiosis 2. This is the end result of meiosis 1. And now we will consider, let's choose a different color, meiosis 2. And, you know, we're not going to be too different. What's going to happen is we're going to reform our spindle fibers. There is no nucleus. That was done away with in meiosis 1. And we have big blue and little blue. just for artistic effect, I'll draw big blue down here and little blue down here. And this is prophase of meiosis 1, where our spindles reform. We don't have to worry about our nuclear envelope, our nucleus dissolving. That already happened. And we don't have to worry about our chromosomes condensing because that already happened. They're already nice and condensed. So we can move on. Well, also, let's do our count. We have 1, 2 chromosomes. Each one is made up of 2 chromatids, or 2 DNA molecules. So 2 times 2 is 4. We can also count 1, 2, 1, 2, 3, 4. And now we move on to metaphase. What does meta mean? In the center or in the middle. Very good. So as I was saying earlier, in metaphase, we try to align our chromosomes together as neatly as possible. And it's so sad. Our chromosomes no longer have their homologous pair. It's so sad. Oh God, it's a little ghost chromosome. They're terribly broken. Not that they're apart. Anyway, no homologous pairs to line up in. So they will just line up in our chromosomes will align in single file, nice and neat. That's metaphase. Once again, we have 1, 2 chromosomes, each only represented by how many DNA molecules? Very good. We know that 2 times 2 equals 4. But now we're getting back into the business. Now, once we're done with metaphase, don't forget that in metaphase, our spindle fibers attach to the centromeres of our chromosomes. And remember, now that we're in anaphase, what is anaphase all about? Anaphase is about simplifying. You have to simplify your genetic information so that you can transition into a gamete. That's the whole point of meiosis. So how can we simplify a chromosome? We can no longer simplify it by splitting homologous pairs from each other. So how can we further simplify this chromosome? Exactly. The only way to simplify this structure is to separate the sisters from each other. And that's exactly what happens in anaphase of meiosis 2. It's that our sisters get split apart. Our centromere no longer holds them together. Think of it like whoosh, they're getting pulled away, whoosh. Because remember, our spindle fibers are contracting. Shoop, shoop, tee, 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 tee. Shoop, shoop. Someone's calling me. Get out of here. Okay. So we have another change in the numbers. So as we articulated earlier, our single squiggly lines, our single condensed DNA is a chromosome in its own right. It is now separate from its sister, and we now consider it a chromosome in its own right. And you might think, they're still in the same cell. Come on. And this one has the same information as this one. Come on. But we know that this left section is going to become its own cell, and so is the right section. So this is the point where we consider them to be different from each other. And so we can count now that we have 1, 2, 3, 4 chromosomes in their own right. And each chromosome is made up of just the one chromatid, just the one DNA molecule. So we've had another change. And then we move on to, hey, please let me, okay, thank you. We move on to telophase. We have our cleavage furrow. We have big blue over here, big blue over here, little blue over here, and little blue over here. And our cleavage furrow forming. Once again, I'd still consider this to be a single cell, and we have 4 chromosomes. Count them, 1, 2, 3, 4. Each one is only comprised of a single chromatid, 4. and 4. What is the relationship of this big blue to this big blue? Exactly, yes, they are still sisters. Just because they were split apart hasn't changed the fact that this one duplicated this one earlier back in S phase. They're still perfectly identical to each other, but they're just separated now. Oh God, once again, it's just so sad. It's so sad. Anyway, moving on. After telophase, we have our final cytokinesis. I'm just running out of space all over the place. That rhymed. T. After this telophase, we move on to cytokinesis of meiosis II. And that is where these cells split apart. So we have one cell and another. We have one big blue, one little blue, one big blue, one little blue. This one became this one became this one. And these are the resulting cells of meiosis II. This cell is now a gamete. It started out here as where'd the pen go? As 2N, and then through meiosis I, it became single N. So what is this now? What is its ploidy? I should have just used the eraser. It's okay. What is the ploidy of this cell right here? Is it half N? Well, we know that N is just the number of chromosomes that represent a specific portion of our genome. And considering that we only had long and short, so chromosome 1 and 2, we have still chromosome 1 and 2, and there's one chromosome representing chromosome 1, one chromosome representing chromosome 2. So we're still just N. We're not half N. It's not like you're dividing by 2 every time, because even though at the end of meiosis I, our chromosomes look like this is haploid because it doesn't have its homologous pair. And therefore, just this one represents the entirety of this portion of the genome. If it had a pair, then it would be 2N. If it had a third partner, it would be 3N. But we have just the single 1N. It is only made up of a single homologous, a single chromatid, but this is still a chromosome in its own right, just as this was an entire chromosome, and we only consider it as one because this one is perfectly identical to this one and provides no new information. So after cytokinesis, we have 1, 2 chromosomes. That should be black. 2 chromosomes, each represented by just a single chromatid, single DNA molecule. Whew. That was a good one. I feel like I missed something. Anyway, oh, that's right. I did miss something. I think Dr. Guntar articulated it already in her lecture, and I highly recommend you watch it if you haven't. Oh, seriously, please watch Dr. Guntar's lecture. She is the professor. She is the overwhelming expert in genetics. I'm just a student, just a cat who talks about genetics sometimes. But yeah, Dr. Guntar talked about some innovations in the cell cycles that allow for genetic variation. And genetic variation is important, especially in meiosis, where we want to create unique daughter cells that each have different combinations, concerts, variations of genetic information. And in metaphase 1, we have 2 possibilities for creating this genetic variation. So let's go back, draw our cell. We are in meiosis 1. Meiosis 1's metaphase. And we know meta means middle. They align in the center. And we have, let's say, big blue was here. And what happens in metaphase with meiosis 1? What aligns in the center? Our chromosomes, obviously. But how do they align in the center? Exactly, in their homologous pairs. They haven't been simplified yet, so they're still the homologous pairs that can align next to each other. So back in the 19th century, a very famous scientist named Gregor Mendel, he analyzed, or he studied pea plants, and we will learn about it in chapter 3, actually. I shouldn't go too deep into it, but he realized or recognized a process during this aligning in the center of chromosomes called independent assortment. And obviously we hope our chromosomes align next to their homologous pair, but why does blue have to be on the left? What if blue assorted to the right? There's no rule that says that chromosome 1, the mother's chromosome 1 has to be on the left side and the father's has to be on the right. The mother's could be on the right. And whether or not this chromosome is on the right is independent of whether or not this chromosome is on the left or right. They assort along the metaphase plate independently of one another. This is, I'm going to try to write nice and pretty. Independent. This is not pretty. I don't know what I was saying. Oh, because I wrote it in crayon last time. So my fault was hidden by the larger font. Anyway, this is independent assortment. And how this creates genetic variation is that we saw that when blue and blue were both on the left last time, what that ended up giving us was a cytokinesis with big blue, big red, little red. But if we have this new condition, our cytokinesis can look like we have big blue on the left, like we see, but little red on the left. And we have big red on the right, little blue on the left. So look at this, the left cell in without independent assortment, or I guess it'd still be independent assortment, but left cell if little blue is on the left versus left cell if little blue is on the right, they are only 50% similar. That is an amazing amount of genetic variation. Obviously, we have 23 chromosomes that each assort independently. So any one, actually, I'm not going to get into that. But yes, this independent assortment in metaphase of meiosis 1 is an amazing potential for genetic variation. It could be just random luck that all of the mother's chromosomes align on the left and all the father's on the right. But that's just due to chance because, chemistry is inherently random, and we mustn't forget that all of our chromosome business is really just chemistry because everything is made of molecules, and a lot of organic molecules are traipsing about and performing their important biological functions. Another introduction of variation in metaphase is when these chromosomes align in the center. Let's zoom in. When these chromosomes align in the center, I'm using the crayon now. Let's just focus on big red and blue. Remember that big red and big blue had the femur length gene at this locus. Oh, I knew I drew that wrong. They have femur length gene. Hey, please come back. Here, at this specific locus, the specific location on the chromosome. And during metaphase, when these homologous chromosomes align in the center with their homologous pair, a random event can happen where this portion of the chromosome switches its position with this portion of the chromosome. Hey, you get over there. And the arms of the chromosome cross over the metaphase plate to exchange a portion of the entire chromosome. And this, unsurprisingly, is known as, crossing over or recombination, or it has a bunch of names. And in chapter 7, we will explore it extensively, linkage and recombination and stuff. And what we would see there is not only independent assortment happening, but we could also have a situation where instead of just having big blue with little blue, we have, and big red with little red, we have big red that's partially blue versus an entirely big red chromosome. And that is just so much potential for genetic variation. You could have so many combinations when you consider crossing over. So yeah, just a little bit of stuff that I forgot. And we will certainly talk more about it. I am happy to talk more about it with you. And so yeah, that's this problem. It is easy to trip up if you don't draw it out. So I recommend you draw it out. It shouldn't take too long. I just took a long time with it so that I could explain some stuff to you. Or maybe not explain, but just give some of my little musings and how I keep everything straight. And I still draw it out all the time. I've been doing genetics for a long time now. So there's no shame in it, certainly. I think it's the best way. But yeah, that is that. That's all I had for you today. I will be around to solve more problems with you, and I should have articulated it. If you go to the LA resources section, once again, you can find the link to a Discord server. And in this Discord server, let me bring it up. Yes, this Discord server is like a kind of community page where you can ask questions about lab or post memes or whatever. You can talk about just the stuff in our class, Dr. Guntar section, or you can petition all of the genetics students at GMU and try to get a quick response. But yeah, it's just a nice little community space for everyone to help each other and get some instant messages and replies in. But yeah, that was that. And I will hopefully see you again soon. Please email me if you need. Also, Yakta and Vaishnavi are available. They're also in the Discord. But yeah, hopefully I will hear from you soon, my friends. Bye-bye.